My name is Jillian Moore. I'm a senior molecular biology major, and I work in the Nachman lab in the Department of Ecology and Evolutionary Biology. Our lab is interested in the genetics of adaptations, so the molecular basis of adaptive traits that increase the fitness of an organism in its environment. And we're also interested in the genetics of convergent evolution, so how two different genetic mutations can lead to the same adaptive phenotype. So one system in which you can study both adaptation and convergent evolution is in the evolution of adaptive melanism in rock pocket mice that live on lava flows across the deserts of Arizona, New Mexico, and northern Mexico. Southern Arizona and New Mexico are where we study these mice. They can also be found to have this dark melanic coat color that matches the dark basalt lava rocks on which they live. So our lab is interested in the genetic changes that have led to this melanic color in these populations. In the Carrizoso population, so this lava flow population, they were able to show that either a mutation in Augoti or in a nearby gene in the genome is responsible for melanism in the Carrizoso population. So we have evidence that the Augoti gene or a linked gene could be where the causative mutation for melanism in Kenzen and Carrizoso is. But the thing that is interesting is that because they don't share the variation, that there are two different changes in the Augoti gene that has led to the same phenotype. So my role in this project has been to obtain more of the Augoti gene sequence in this rock pocket mouse species, and to sequence the regions that I'm able to obtain in light and dark animals, and look for differences that are associated with coat color to try to identify a causative mutation. After many months of molecular biology work, I have been able to piece together the fragments that they had before, expand the sequence we had before by a couple thousand base pairs. The technique I use to do this is called genome walking. So it involves cutting your genomic DNA up into pieces using restriction enzymes. And on the ends of these pieces, you ligate adapters, which are just strands of a DNA sequence, and you know this sequence. But the sequence stopped where the restriction enzyme cut the DNA. So I'm currently redoing the experiment using different restriction enzymes that hopefully do not cut in the middle of the coding sequence I'm interested in. And the second thing I've been doing is, of course, sequencing these regions I obtain in light and dark animals. So far I've sequenced 15,000 base pairs in 2 light and 2 dark animals from this Carrizoso population. So far I have not found any variation that is associated with coat color, but we are continuing to sequence the regions we get through the other molecular methods we're using. And we have also just recently sequenced the genome of this organism, so hopefully we can search through this sequence using the sequence we do have to find more of the Augoti gene. And in the future, we hope to find the mutations responsible for melanism in the Kenzen and Carrizoso populations to better understand how convergent adaptations arise in populations under similar environmental pressures. So thank you.